So stop codon readthrough is the process when a ribosome, instead of terminating at a stop codon, incorporate an amino acid in the position corresponding to that stop codon. And we know that it occurs in different species, and particularly more often in certain viruses that use this process to regulate their translation and protein synthesis, because, say, stop codon readthrough would not be a very efficient process. So normally it's only a small fraction of the ribosomes that would incorporate the amino acid. And because of that, say, you could have two open reading frames separated with a stop codon, and then the product encoded in the first open reading frame will be translated and synthesized in far higher rate in comparison with the downstream region. But in addition to viruses, stop codon readthrough is also found in other species. In particular, for not well understood reason, it's quite often, quite frequent in the translation of flies and mosquitoes. But in humans, certain genes also use stop codon readthrough in their expression. Interestingly, one of the most efficient cases of stop codon readthrough in humans occur in the opioid receptors in our brain. And in some cases, what it helps to make in the cell is proteins that would have different C-terminal ends. So one would be longer than another, and this extension, for example, it could be used, for instance, as there are a number of theoretical possibilities, but for instance, it could be used for signaling transport of a protein into a particular compartment in the cell, or for secretion or something like that.